Chapter 8 is titled The Cellular Basis of Reproduction and Inheritance. All types of reproduction can be classified as either asexual or sexual. Asexual reproduction is when you have one parent that produces two identical offspring. The offspring are identical because they get the DNA from just the one parent. This is a picture of an amoeba that is a single-celled organism dividing in half to produce two identical offspring. With sexual reproduction, two parents combine their DNA to make offspring with a unique combination of the two sets of DNA. So with these two parents, they are each going to contribute half of their DNA to their three children. Now these three children, these siblings, they do not have the same DNA because which half of the DNA they're getting from both parents is going to change. Sexual reproduction, it does require more energy to produce egg and sperm, for example, but the huge benefit is that it produces variation, and variation is very important to evolution. The cell theory states that cells arise from pre-existing cells. So cells don't come out of nowhere, they have to divide in order for you to get new cells. The purposes of cell division. Okay, one is for reproduction. We already covered that. Both sexual and asexual reproduction, they require cell division. Cell division is also important for growth. So when organisms are growing, they are having their cells divide in order to make more cells for the organism. Cell division is also for repair if there are damaged cells. Also just renewal. Cells have a certain timeline where they live for a certain amount of time, and then they go through apoptosis, which is programmed cell death. Those cells that are dying, they will be replaced by cell division from the surrounding cells. Prokaryotic cell division is called binary fission. It is an asexual process because we just start with one parent. It's pretty simple because prokaryotes, they lack most organelles. They only have ribosomes. They don't have a nucleus enclosing their DNA. One thing, too, that is unique about prokaryotic DNA is that their DNA is circular instead of having linear chromosomes like eukaryotes do. I know this doesn't really look like a circle, but their DNA, it's really one long circle, and it does have to be kind of crumbled up in order to fit in the cell. So this is really like a crumpled up circle. There's three basic steps to binary fission. The first step is that the chromosome has to duplicate, and then the copies are going to separate. Step 2 is that you have continued elongation of the cell. So the cell is going to elongate and the movement of the copies of the DNA. And then finally, you have division where the cells are actually going to split, and you're going to have two identical daughter cells. Daughter cells are just the cells that result from cell division. Eukaryotic cell division is more complicated. That's what we're going to focus on for the rest of the time. Eukaryotes have linear DNA, so it's not circular, and it's wrapped around proteins. The proteins help organize the DNA. Chromatin is what we call DNA in the loose state. So the DNA is wrapped around proteins. It's loosely wrapped. Something that has to happen before cell division, the DNA does have to be duplicated, just like in prokaryotes. But then also this loose DNA, the chromatin, it uses the proteins to condense into a tighter form of organization called chromosomes. Chromosomes then are DNA wrapped around proteins. This is an electron microscope picture of a chromosome. It's very compact. The DNA is tightly wound around these proteins. Chromosomes are often depicted as just these lines, okay? Because chromosomes, it's linear pieces of DNA that are wrapped around protein. One chromosome is going to be representing one piece of DNA. When the chromosomes duplicate, or when the DNA duplicates, now I have these two chromosomes, or they're now two identical pieces of DNA. I can call these sister chromatids. They are held together in the middle by this place called a centromere. But since these sister chromatids, they originated from the same chromosome, they're identical. The whole purpose of copying chromosomes is so that during cell division, the chromosomes can be distributed to the daughter cell so that the cells have the same DNA that they did in the original cell. Before we talk about specifics with cell division, we are going to mention some equipment that's needed. One piece of equipment that's needed for cell division are spindle fibers. Okay, these are these spindle fibers. They are made of microtubules, which is a type of cytoskeleton. Cytoskeleton literally means skeleton of the cell. It gives cell structure. It allows movement of the cell and things within the cell, just like our skeleton. These microtubules, they guide separation of sister chromatins. So they're going to be the things that actually grab onto the chromosomes and move them around the cell during cell division. Centrosomes, so we have two right here, these are where spindle fibers emerge from. If you look inside of them, there's two little structures. Those are known as centrioles. Those centrioles are actually only found in the centrosomes of animal cells, not plant cells. Eukaryotes have the cell cycle where they're doing a lot more than just dividing. And actually, the division part of the cell cycle should be pretty small. Most of the life of a cell is spent in what's called interphase. Interphase is made of three parts. The first part is called G1, or the first gap. Something that's happening in G1 is the cell is growing and it's carrying out whatever the specific functions are for that cell. Then there's the S phase. The S is for synthesis. Specifically, what is being synthesized is more DNA. So this is actually the phase where DNA is going to be copied. Then there's another gap phase where the cell is not dividing, it's not duplicating its DNA, it's just growing and it's going about its normal cell business, carrying out whatever its cell function is. Then there's the mitotic phase where cells are actually dividing. The mitotic phase, or M phase, is made out of mitosis and cytokinesis. By the end of interphase, cells are now preparing for cell division. They're preparing for mitosis and cytokinesis. So the cell should be doubled in size so that after division, you don't have cells that keep getting smaller and smaller. You should have two centrosomes, and you'll see why. And the chromosomes should have duplicated in the S phase. Now, if I actually look, I cannot tell that the DNA has been duplicated because the DNA is in this loose chromatin form. That's why it looks like spaghetti. If I look at this cell at the end of interphase under the microscope, in the nucleus, I cannot see the DNA when it's in this loose chromatin form. Now we're going to start going through the phases of mitosis. The first phase is prophase. In this phase, the loose chromatin is going to condense into tight chromosomes. So now I can actually see the chromosomes. I can see how they consist of sister chromatids. When DNA is in this condensed chromosome form, I can now see it under the microscope. Something else that's going to happen is the nucleoli will disappear. Nucleoli are sites in the nucleus where ribosomes are being assembled. I can also see that the mitotic spindle begins to form, and my two centrosomes are beginning to separate. Second phase is prometaphase. In prometaphase, the nuclear envelope breaks down, and now these chromosomes can be moved wherever in the cell. The microtubules, they are connecting at the centromere, which is the middle of sister chromatids. And at the sister chromatids, there are these kinetochore proteins. That's actually what the microtubules connect to. In my microscope picture, I can see these blurs right here. Those are the centrosomes. Next phase is metaphase. By metaphase, the mitotic spindle, it should be fully formed where the centrosomes are on the opposite sides of the cell, and then all the mitotic spindles, they're connected either to each other in the middle or they're connected to the chromosomes. Metaphase, this is when chromosomes line up in the middle of the cell. the middle of the cell, there's this imaginary line called the metaphase plate. I can see the chromosomes lining up in the middle of this microscope picture as well. Next up is anaphase. With anaphase, the duplicated parts of the chromosome, or the sister chromatids, they're going to separate. They're going to move to opposite poles, opposite sides of the cell. At the kinetochore, there are these motor proteins that basically walk along the microtubules. These motor proteins are very similar to the motor proteins in our muscles that make us move. The spindle fibers that are connected to chromosomes, they are going to shorten in order to reel those chromosomes in. The spindle fibers, you can see some of them, they're actually not connected to chromosomes. They're not going to shorten, they're going to lengthen in order to elongate the cell. Telophase is the last phase of mitosis. You can see a lot of stuff that happens in telophase is actually opposite of what happens in prophase. The cell is going to continue to elongate. The nuclear membrane is going to reform around the chromosomes. The DNA is actually going to relax. So the tight chromosomes, they're going to unwind into loose chromatin. The nucleoli are going to repair so that my cells can start making ribosomes again. And then the mitotic spindle, since I don't need that anymore, that's going to disappear. Cytokinesis is not considered a step of mitosis. It does start occurring at the same time as telophase. What cytokinesis is, it's the division of cytoplasm and the complete separation of cells. So at the end of cytokinesis, you have two identical daughter cells. Now, cytokinesis does happen differently in animal and plant cells. In animal cells, I have this cleavage furrow start to form. The cleavage furrow, it is made of a contractile ring of microfilaments. Microfilaments are a different type of cytoskeleton. It's kind of like a drawstring that starts closing in to pinch the cell off in the middle until you have a complete separation of the cytoplasm and two distinct daughter cells. Now, this happens differently in plant cells because plant cells are just separated by cell walls. So what I have starting to happen at plant cells is that vesicles will collect in the middle that contain material to build the cell wall. That material will start to be put together and I'll form a cell plate. And then finally that cell plate will elongate all across the middle of the cell. I'll have a new cell wall built in between my two plant cells. Cells have a lot of mechanisms to control cell division because uncontrolled cell division can lead to cancer. So one way cells do this is with anchorage dependence, and basically this means that cells have to be anchored to something in order to divide. That can be a surface like in a petri dish, or it can be a surface of the extracellular matrix. Extracellular matrix is just molecules outside of cells. Another method of controlling cell division is density-dependent inhibition. With density-dependent inhibition, this is when cells have formed a complete single layer. They will stop dividing because they are all touching each other. Now, if some cells are scraped away, the remaining cells, they will divide in order to fill the dish with a single layer again, but then they'll stop dividing. So this, for example, is what happens when you have a cut on your skin. Cells also control the rate of division by producing growth factors. These are proteins that stimulate cell division. Now, they should only be produced when cells need to divide. What this picture is showing is that if we do have a single layer of cells, but then additional growth factor proteins are provided, then the cells are going to start dividing. So further cell division is going to be promoted. Another way that cells control the rate of division is by these checkpoints. These checkpoints, they occur along the cell cycle, and what they do is they respond to signals, both what's going on outside the cell and what's going on inside the cell, to make sure that everything is happening correctly. The first checkpoint, there's one that you can see that's towards the end of G1, so there's this G1 checkpoint. There's another one right at the end of G2, the G2 checkpoint, and then there's another M checkpoint that happens right in the middle of M phase, which is mitosis and cytokinesis. Cells will pass this G1 checkpoint really if growth factors are present in their environment, and that's giving the signal for cells to go ahead and proceed through the cell cycle, which will end with cell division. So these growth factors, they are going to bind to receptors on the cell membrane. The receptors are going to relay signals inside the cell and tell the cell, okay, go past the G1 checkpoint, continue on with the cell cycle. The G1 checkpoint that has the stoplight on top of it seems to be the most important in cell division, meaning that if cells are able to pass the G1 checkpoint, typically they are able to pass all the checkpoints and proceed through the cell cycle. If cells are not receiving the growth factors in order to pass the G1 checkpoint, they can enter this G's of 0 state. The G's of 0 is a non-dividing state. It could be permanent, it may be temporary, but it is permanent typically for mature nerve and muscle cells that are in a non-dividing state. The situation where it would be temporary is if finally growth factors do appear in the environment and then the cells can pass the G1 checkpoint.